What is SCID? SCID. It stands for severe combined immune deficiency. It is a hereditary defect in which infant lack antibody formation. Antibody is not formed due to impairment of component of immune system, that is the T lymphocyte and B lymphocyte. Children suffering from the SCID can die even an ordinary disease, vaccination and blood transfusion. In 25% of SCID cases, patients have a defective autosomal gene for the synthesis of enzyme adenosine deaminase ADA. In short, it is known as the ADA. In absence of ADA, purine metabolism is disturbed and T lymphocyte fail to function. ADA gene is taken out from the leukocyte of a healthy person, amplified and inserted into retrovirus from where viral gene have been taken out. A helper retrovirus is also prepared divide of packing sequence. Lymphocyte cells are taken out from bone marrow of patient and injected into two type of the retrovirus, one carrying ADA gene, another is helper 1. The gene begins to express in the lymphocyte. The cells also possess faculty gene, but also the effect is now suppressed. The transformation cell, which are healthy stem cell having functional ADA gene, which are introduced into bone marrow of the patient, the immune system become functional. Next one is the gene therapy. It is a therapeutic treatment of defective heredity by introducing of normal, healthy and functional gene which silence the defective genes of individual. Gene therapy should be applied to germ cells, especially zygote, so that gene progeny gets rid of the defect. Gene therapy is done in few cases like the SCID, cytosis, fibrosis, atherosclerosis, thalassemia, muscular dystrophy, and several other diseases. In gene therapy, a desired gene is isolated, amplification, a retrovirus lacking packing sequence gene and inserted desired of the other sclerosis by retrovirus lacking packing gene. Next topic is the trangenic. Any organism containing a foreign gene from a different species is a trangenic organism. A gene can be transformed to the genome of another organism only through the genetic engineering definition. Genetically engineered organism carrying gene of another species in their genome. Transgenic organisms are also called genetically modified organism. In short, it is known as the GMO. GMO is created by incorporating into their genetic makeup foreign gene or extra copy of an endogenous gene or clone or modified. Transgenic crop can be produced through a genomic character. For example, the gene coding for the insecticidal protein from Bacillus thuringiensis has been transformed to the cotton plant. This transgenic cotton, known as the genetically modified cotton BT, is resistant to the bollworm insect. Another example of the transgenic plant is the tomato. The GMO tomato called flavor saver and much longer and blended with flavor than the conventional tomato. because of delayed ripening. This is achieved by reducing the amount of the cell wall regarding enzyme polygalactoronase responsible for fruit softening products. Production of gene high value of pharmaceutical important compounds such as the human insulin, interferon, hormone, and blood clotting factor is transgenic plant or seeds. Production of antibodies and vaccine is transgenic plant like the banana and tomato. Children may get immunized against the several diseases like the cholera by eating banana or raw tomatoes from vaccine. What is genome? All the DNA in the cell of an organism called genome. All the DNA in a cell of an organism called genome. First human colon is done in 26 December 2002. Baby girl called Eve, surrogate mother. When normal or altered egg is planted in a different female is called surrogate mother. Thank you.